All right, Microbiology 221 students, today is day 3, and today is probably the longest day. There are several ways to divide it up so that you only have to do about 45 minutes, half hour, 45 minutes of work at a time. So there are some ways to divide it up. What I would suggest is the following. Today we're going to do staining. We're going to prepare some slides that we can stain. So we need to first make smears, and those smears need to dry, and then we're going to fix them using our little paraffin burner. And then we need to stain those smears, and then we need to view the smears under the microscope. So I would suggest dividing it up into those 3 parts: making the smears and letting them dry, fixing them. Once they're fixed, you have to do that in the same day, because once they're fixed, then you can stain them tomorrow if you want, or 2 hours later. Then you have to go through all the staining procedures at once. I would suggest, I'm going to show you doing the simple stain and then also the gram stain, but I am going to make all my smears at the same time for both of those activities. They would be activity 1 in the simple stain, it's activities 1 and 2, and activity 3 is actually the gram stain or the differential stain. So we're going to make the smears, dry the smears, fix them, and then I'm going to go through the staining procedure with you for both a simple stain and a gram stain. And then we can put them aside, and you can look at them under the microscope whenever you have time at whatever pace that you want. You can view them, draw them, and then if you need to repeat the experiment, you can repeat the experiment, clean off your slides, repeat the experiment, and look at them again another day. The second thing that we need to accomplish today is we need to take a look at our results from yesterday to see if everything's grown up enough that we could record our results. My results did not grow up well enough. So I can see a lot of growth in my broth culture, and I should probably put that behind a dark background, which I don't happen to have with me right now, but yeah, not too great. But you can see that it's cloudy, hopefully. So the broth cultures look great. The slants look pretty good, but the cultures are a little bit light. And my Petri dishes for both my just streaking serratia, which I think you can see growing on there, and my mixed cultures, they did not grow enough. My mixed cultures, I only have growth up here in the first quadrant. It's rather cool up here in where I'm doing my work. So I'm going to let these just incubate until tomorrow. And I'm probably going to move them to a little bit warmer spot so that I can take a look at them tomorrow and record those results. I am going to wear gloves today just because we're staining and I don't want the dyes all over my hands. If you prefer not to wear gloves for the staining procedure, or if your fingernails cut through the gloves and you do get stain on your hands, I would recommend the following stain removal procedure. I would use a light cleanser, like a soft scrub, or some Bon-Ami, something that's not super abrasive. And I would try to use it dry. You'll get most of the stain off. And then of course you can use rubbing alcohol. Will probably remove most of the rest of it. After a couple days, it will go away as well. So I wash my hands before I put the gloves on. And I have my bleach solution here, which I'm going to use to clean off my work surface. And again, I probably should have done this prior to laying out all my tools, but I was in a hurry to try to find everything. So I wanted to be sure that I had all my materials. My kit did not come with a methylene blue stain. I don't know if yours did or not. I think it was supposed to. So if you did not get the methylene blue in your kit, you can just skip that simple stain. That's what I'm going to do. I'm going to skip the simple stain for methylene blue and just use the crystal violet and the saffronin, which are part of the gram stain set. So aside. So I have my cultures here in my little test tube rack. They're labeled. Remember, we rehydrated 2 of them a couple of days ago, and then we rehydrated the E. coli and the staph epi yesterday. And they look well grown up in order for us to be able to take a look at them, as well as I'm trying to find a piece of construction paper. Anything that's a dark background, they will show up a lot better for you to see. If I can find something that's black to put them up against. Yeah. Okay, so I'm out of luck here. All right, so I can't show you. The paraffin candle, we want to be sure to keep that away from the denatured Isopropanol, because that's 95%, it will light on fire. I believe my stain set came with 2 bottles of that. So I'm just going to keep that a little further back. And you might notice that I've arranged my reagents here in the order that I'm going to use them: crystal violet, the grams iodine, the denatured alcohol, and then the safranin. Both the crystal violet and the safranin came in dropper bottles. The other 2 did not. So I'm going to insert a sterile pipette from my sterile pipette collection. Came in my box. And I would give these a little shake before you put the pipettes in them. My grams iodine leaked too. When I pulled it out of the box, I noticed that it had leaked all over the place. So I'm gonna just set that aside and put the dropper bottles and eyedroppers in there so that when I go to reach for them, I am all set and ready to go. And what I did notice is we need to rinse frequently with distilled water, and they did not give us a bottle of distilled water. So I have my big jug of distilled water here, and I'm looking around in my kitchen cabinets, and since it's just distilled water, I think it's okay. I'm just going to use a teacup, I think, or maybe a glass. Let's find a glass. All right, all I have are these from my grandmother wine glasses here, little cordial glasses. So I'm just going to put my distilled water in there because that's what I happen to have. You're more than welcome to put it in a beaker or another one of these plastic cups. That's probably what I should do, is go downstairs and get a plastic cup, but I'm kind of in a hurry, so I'm just going to do that. All right, I'm going to stick the rest of my distilled water back here in the back. All right, myself, I would prefer, your lab book asks you to stain them over a sink. I'm going to use a little staining tray. I just have a small, Rubbermaid, or a small, I can go from Target actually, a little container for food storage. And I happen to have 2 flat little pieces of wood. I can set those across there like my as a staining tray, so that I can just put my slides on there and stain without having to do it over the sink. I intended, and I didn't find a pair of scissors up here, so I intended to use this larger staining tray and just notch out some plastic straws so they wouldn't roll around on there. I was going to use that too. So you might have to come up with a little ingenuity for this portion. I think for the first, for our simple stain, I can probably just get away with this because I'm only going to have 2 slides instead of 3 because I don't have any methylene blue. All right. Get out a couple slides, put the rest of the stuff to the side. I would strongly recommend a couple of other things. I have already written out the procedure on some paper over here, like as if it was my lab book, and I have that where I can easily see it. You're going to need a timer of some sort. I'm going to use my watch with a second hand because I don't want to touch my phone with my hands after I've been handling bacterial cultures, but that's totally up to you. You can use your phone. I would set the timer for a minute, and then you'll be able to just kind of reuse it over and over again. And again, if you touch it with your pinky and your pinky hasn't touched any bacteria, it'll probably be fine. But I'm more comfortable just using my watch. So I'm just going to use my watch. Now, they recommend labeling the slides. And as I said, since I don't have the methylene blue, I'm only getting out 2 glass slides, and they do recommend cleaning them. I'm not going to do that. I'm going to skip that step. And I'm going to lay them right down here on my lab bench. And I am not going to use the China marker because we're not going to use any alcohol with these 2 slides. So I'm just going to draw a couple of circles. They ask you to label this slide 1 and A for your first circle, B for your second circle. So like that. And then the second slide, I'm going to do the same thing. I'm going to label it slide 2 in the lower left-hand corner. I'm going to draw 2 fairly large circles. And again, label one of them A and the second one B. This will help for how you need to draw the pictures in your notes. I would also, because I'm going to make all the slides and all the smears at the same time, I'm going to label this one up at the top, SS, for simple stain. And then I'm going to label my other slides GS for gram stain. Okay. And again, you're welcome to use the China marker. I find them harder to write with. I would just run to Target or into a hardware store and I would get myself a Sharpie because they don't come off unless you use alcohol in between, which we will use for our second. So again, I'm just going to use this for my staining procedure and I'm just kind of checking out how it's going to work. It looks like it's going to work fine. And then I'm going to kind of set this aside. I believe I am going to have to light my paraffin burner and I forgot my lighter. So we're going to take a short pause here where I'm going to stop recording and run downstairs and pick up my lighter. Oh wait, can I pause that? Let's see if we can just pause. Is that a All right, I think we're back in business. So I did go downstairs and pick up my lighter as well as just a plastic cup, and I labeled it DH2O for distilled water rather than using that microscopic wine glass. And then again, I'm going to light my paraffin burner because I am going to need to flame these tubes as I collect bacteria out of them and put them on the slides. So the directions recommend, and I think this is, good practice, good lab practice. Speaking of which, please put your lab apron on, wearing closed-toed shoes in case you drop something. And it would be probably best if you used your safety goggles. I'm wearing my glasses, so my reading glasses, so I'm taking that to be good enough, but it's up to you. So the first thing you want to do is when you have your two slides, I prefer to lay mine right on the lab bench, not on a piece of paper or paper towels, because it does tend to make the work surface a little bit unsteady. It's totally up to you. If you're more comfortable with having a paper towel, I would do a single layer of paper towel just in case you had any problems with things dripping over. So on each of my slides, I'm going to place a single drop of water, of distilled water, and I want this drop to be relatively small because the smaller the drop of water, the faster that the slide's going to dry. So I am putting, and I'm just doing this on purpose, I'm showing examples of, I can bring the camera over so you can see. Right on that. So I have some small drops of water and then some that are large drops of water. But again, the smaller the better as far as the experiment goes. All righty. Next, we need to put the bacteria on there. So again, I'm going to use some of my sterile pipettes that came in my kit, came in my big Carolina box. And actually, we're not going to use a pipette. We're going to use a inoculating loop. Sorry. So I still have this pack that I was using from yesterday. So I'm going to do the same procedure that I did yesterday. I'm going to try to get one inoculating loop all by itself, close to the surface, close to the Ziploc tab, so that I can open up this very carefully and withdraw a single loop. I'm going to start, I've already made on my notes, I've already made which sort of a reminder to myself what the order is going to be on each of my slides. Slide number 1, circle A, I believe it needs to be E. coli. So I'm going to take my E. coli tube, give it a little gentle swirl, remove the cap with my pinky by turning the tube, flaming this top of the tube very gently, taking my loop, popping it in here, again, trying not to touch the side walls, removing a loopful, flaming, closing by turning the tube, not the cap. And then I'm going to put the E. coli back here close to my cultures I need to read tomorrow, so that, again, I'm working from the front of the rack to the back of the rack as much as possible. And then I'm just going to simply mix my E. coli in my A circle with the distilled water that I already put in there. It's important that I just mix like this, no smacking. Smacking will put the bacteria into the air and we don't want that. Like I said, I like to be right on the lab bench myself, so I'm going to do that. And tap a little bit And then again, this is still full of, potentially full of E. coli, so I'm going to drop that into my ethanol jar. Now, letter B is supposed to be the staph epidermidis, so I'm going to withdraw another single loop here and get my staph epi container, my broth culture from yesterday, give it a little shake, a little swirl, so the bacteria are all nice and resuspended. Twist the tube, flame the top, almost forgot. Insert and get a loop full of bacteria, flame the top, cap the tube, put the tube down, again, moving it to the back of the rack and placing it on slide number 1, circle B. And I'm giving that a good mix. Let's smear a nice little mix. And then I'm going to pop my loop into the isopropanol. So slide number 1 is finished. It just needs to dry. I'm going to set that aside. I'm going to move slide number 2 off of my paper towel here and get the bacteria on slide number 2 here. So that is going to be in letter A. Circle is going to be the Serratia. So I'm going to tighten that cap, give it a little shake. Ooh, there's a lot in there because this is day 2 for the growth of the Serratia. I'm going to flame the top of the loop, top of the tube here. Reach in, grab some bacteria, and mix those around in circle A. Flame, close, move that to the back of the rack, deposit that in my waste container. One more. Get out a sterile loop. Pinch the bag shut. The last one is Micrococcus luteus. So this grows the most slowly, and I don't know, you can probably tell amongst your own cultures that this one is a lot less turbid or less cloudy than the others. So I'm trying to be sure that I give this a really good shake because the bacteria are probably set kind of in the sediment at the bottom of the tube. Unscrew the cap by unscrewing moving the vial, not the cap, flaming the top, inserting, picking up a loop full of bacteria, flaming, closing by turning the tube, replacing the tube in the rack, and this goes in my last circle here. Okay, excellent. And then I'm going to pop that right into my isopropanol jar. I am going to see here, I can hold my hand about this far up. So I am going to warm my slide a little bit to help it dry, but I think I'm going to pause our video for now because we can't continue until these slides are dry. So I'm actually, changed my mind there. I'm going to set these aside. They're SS1 and SS2. And I am going to go ahead and prepare the slides for the gram stain as well. So your instructions are very specific for the gram stain slides. They require you to make 4 slides so that you have all the different combinations of bacteria. So I'm going to get out 4 more slides. And the last slide, slide number 5, is going to be a tooth and gum scraping. So we're actually going to set up 5 slides. And I realize in your lab booklet, they make this 5th slide a whole other activity. Our lab kit did not include a pre-gram stained prepared slide for you. So you're only going to have 5. You would, if it included that extra slide, it would be here. It would already be stained. I would also advise getting a Post-it pad and writing down the procedure for the gram stain in shorthand, which I have on my paper here, how much time for each stain. And then I would post this like right in front of me so that all I have to do is look up when I'm in the middle of my procedure and not fuss around and look how long do I leave each thing on, each stain or each decolorizer on the slide itself. I would advise to use the crayon for this slide, these slides. And the reason for that is because the alcohol in the decolorizing step will remove the Sharpie. Now, in lab, we normally ask students, we tell them to put, you're going to put 2 circles on each of these slides, except for number 5, because that one you're going to just have a single smear. And then we usually indicate to students it's wisest, it's best if you actually flip the slides over and put your bacteria in those and use them just as target circles. If I were you, once you flip the slide over, I would number my slides 1, 2. This is tough. I think I'm going to use the Sharpie anyway. 1, 2, because it actually writes. 3, 4, and actually, no, you're supposed to label them 1, 1. My apology. 1, 1, 2, 2, and then this is, I would just label this one tooth. And you'll remember because it only has one circle on it. And then you are supposed to label them A and B. So 1A, 1B, 1A, 1B. Two A, 2B. And then in your notebook, myself, I would write down which bacteria you're going to call A and B and C and D. You know what? I might actually recommend a different way to do that, but for right now, we'll do it this way. We'll do it like it says in your booklet. So what I have written down that we need to first put E. coli together with Serratia. So I'm looking for E. coli and Serratia, or sorry, E. coli and Staph epi is my choice. I think it tells you can do them any order that you want. I'm going to put E. coli with Staph epidermidis on slide number 1, and then I'm going to put E. coli with Micrococcus luteus on slide number 2. Again, like I said, you can do it in any order that you want. I would put, again, small drops of distilled water. And if they don't land perfectly, that's fine. And again, we're going to need loops for each of our transfers. So I'm going to put my E. coli on all the slides, which for me is going to be slide 1 and slide 2. My paraffin burner does not stay lit very easily. Like if I talk, it goes out. So I'm just doing it this way to save some time. So I'm going to put a loop full of E. coli, slide 1 in the A spot, mix, mix, mix. And then I'm going to put a second loop full of E. coli on slide 2 in the A spot. And then I'm going to flame. Myself, I think it would be wise of you to label these with the initials of the bacteria underneath. So I'm going to put EC under both of those. And then I'm going to put my Staphylococcus epidermidis. I'm going to put that on 2 slides as well in the B position. My slide 1 Staph epi and my slide 2 Staph epi B. So then I will only have to use, I'll use fewer loops this way. So I removed a loop sterilely, aseptically, flamed the top, and I'm going to grab some Staph epi B, put that there, and another loop full for slide 2B. That doesn't look like it had any water on it. I'm going to give that a second loop full. All right. Close. Put that down. Pop that in the container. All right, so now I've done the E. coli, the Staph epi. Now I'm going to do Serratia. And I put the Serratia on my slides. Slide 1, my second slide 1A position, and my second slide 2A position. So again, I'm only going to have to use 1 loop. And I will go to the whiteboard while these are drying and put the order that I put my bacteria on for you so you guys can see. the Serratia is tons of it. So slide 1, letter A is going to be Serratia. And slide 2, letter A is going to be Serratia. There we are. I'm going to flame the top of that, pop that on, stick it in there. And now I believe I should only have Micrococcus luteus left. And I'm going to use second to last loop from bag number 1 here. And Micrococcus luteus is going to go into the last 2 that aren't smeared already. So Micrococcus luteus, I'm going to put that here in B. And I'm actually going to put 2 loopfuls on there because it didn't grow as well. And Micrococcus luteus on my first of slide 2B. And again, I'm going to put 2 loopfuls on. Try to get 2 loopfuls on mixed in there. Flame, close, put that in there. And then I'm going to hit the whiteboard and I'm going to tell you a minute how I loaded those. Oh, and we should do the toothpick one while we're here. So I did get a toothpick, and you want to, no blood, okay? So you want to just go kind of in these gingival cavities, kind of up here where your teeth meet your gums, like right there. And then you're going to smear that into the water. And we're going to see what kind of bacteria we have in our mouths. Now, technically, this is full of bacteria. You can just throw it in the trash. I am going to pop mine into the ethanol container. Now, I want to real quickly go to sharing content with you, and I'll jump down here to a whiteboard. So my slides, how I set them up, just so you guys can see real quickly, is this is for the gram stain. So they told you to make 2 slide 1s and label them A, B, A, B, and 2 slide 2s and label them A, B, A, B. And you know what? You can skip the letters if you want to just put the names of the organisms. So they're having you make 2 sets basically in case you have a mistake. So my set number 1 here, I put E. coli in that A circle for my smear. And then in my second one, I also put E. coli here like this. In my B, I put Staphylococcus epidermidis. And then I also put Staph epi over here. I put Serratia here in letter B, I believe. No, sorry, Serratia in A and A on these sets. So this was Serratia and this was Serratia. Serratia marcensis, so SM, capital S, lowercase m. And then for Micrococcus luteus, I put Micrococcus luteus here, capital M, little l, and here. Okay, so each set, what's important to note is each set, like for slide 1 here, I have 2 slide 1s and I have all 4 bacteria on 2 separate slides. And I actually, and there's a table in the lab handout that has calls them group A and group B. And basically we're putting 1 gram negative with 1 gram positive, 1 gram negative with 1 gram positive. And then in my second set, in case my first set doesn't turn out very well and I have to repeat the gram stain, notice I have all 4 of them, but I've switched their partners. So this time, instead of putting E. coli with Staph epi, I've put E. coli with Micrococcus luteus. And instead of keeping Serratia marcens with Micrococcus luteus, I've switched it with Staph epidermidis. Okay, so this is my setup for the gram stain slides. And then we also did a slide, all right, of tooth and gum scraping, and that was just water and a toothpick. All right, so I'll draw a little toothpick there. And I'm not going to label this anything because there's only one circle on there. There's only one bacterial smear on there. All right. So I don't need to label it because it doesn't really matter. All right, so I'm going to stop the sharing and try to reset up my camera here. And these slides are not dry. So I'm going to pause the recording and we'll come back when they are.